evening, good morning, or good afternoon, depends on where you are. Hello, everyone. Welcome to ICANX Talks 141 series, Connect the World and the Universe. So, my name is Rong Chen from Huazhong University of Science and Technology, and I'm going to moderate the talk for tonight. And today, it's very fortunate that I have a co-moderator with me today, Martin, that's your old friend from North Carolina State University. But today, he is in UBC. And as you can see, we have a wonderful and fully packed April. And we start with the Young Scientist Award, and then we have Professor Xue Feng from Tsinghua University and Professor Sarah Bells from University of Antwerp. And today, we are very fortunate to have Professor Beomjoon Kim from University of Tokyo. And today, we have a very strong panelist as well. And we have Professor Mark from Georgia Institute of Technology, and also Professor Boris from UBC as well. And we have our ex-challenger, Jiayan Zhang from Peking University. So first, let me introduce Professor Kim from University of Tokyo. Currently, he is a director of the LIMS Kiko and also Center for Research on Engineering in Medicine and Biology. And Professor Kim is in charge as a chair of a corporate sponsored research division of virological medicine of University of Tokyo. Professor Kim received his bachelor from Seoul National University, master and PhD from the University of Tokyo. And Professor Kim investigated several aspects of biosensors components to accomplish portable point-of-care diagnostic devices, which are disposable, user-friendly, and low-cost, and highly sensitive. He's interested to develop self-powered energy harvesting microsensors as well as smart monitoring systems. Professor Kim has published over 100 peer-reviewed journal papers, more than 200 international conference papers, and more than 200 domestic conference papers, as well as tens of patents and books publications so far. So without further ado, let's welcome Professor Kim to the stage. And tonight, his talk is biomolecular metering system for medicals. Professor Kim, please come to the stage. Thank you.
Okay. So can you hear me?
Yes.
Okay.
And we can see we can see your slides as well.
Okay. 谢谢. So hello everyone. So 你好. Good evening or good morning and good afternoon as well. So thanks so much again for Professor Long Chen for a nice introduction of me. And first of all, really appreciate so much again for giving me this very honorable chance to introduce our research work here in great ICANX talk with the very famous professors as well as a pioneer of microneedle research field who is Mark and Boris. So I'm actually very nervous now. Well, so okay, so today my talk is related with MAP, that is microneedle array patch, beyond the skin barrier. So to reach transdermal drug delivery as well as self-testing diagnostic biosensors. That is today's my talk. So I go to next slide. So what is better for your drug delivery system? So you can see here is a 4 kind of drug delivery system comparing with the hypodermic injection or this oral inoculation. So which are more than 80% of the whole drug delivery system nowadays. But they're comparing with these two methods. So you can see here, drug patch or ointment way are the most convenient or painless and daily usable and local treatment of a drug delivery system. But however, due to, as you know, is already known very well, but due to the skin barrier of our sternum corneum, these are really our protect our body from the outside environment. Therefore, this sternum corneum barrier, these conventional drug patch or the ointment, of course, normally normal cosmetic as well, cannot deliver the actual API, which is active pharmaceutical ingredient, which are more than 500 dalton of high molecule weight, cannot penetrate actually. Of course, something maximum 30% can diffusing and penetrate, but almost of this more than 500 daltons of molecular weight of the high molecule cannot penetrate through the skin barrier. So that is our motivation of the research for micro needle array patch. So what is micro needle array patch? That is, yes, you can see now here it's a mosquito. When a mosquito bites you, it doesn't hurt, but after the area will becomes just itch later, right? But this is because the tip of the mosquito, the sharp and straw like a mouse, can pierce the skin. It's about 60 micrometer in diameter. That is the main reason you cannot feel any pain. The even tiniest the metal injection needle available for insulin injection, for example, is outer diameter is almost 180 meter, 180 micrometers, so which can be painful still. Also, every day there is some burden for the patient, right? So, well, just the next slide. Therefore, ですね。はい。So, but it is Japanese, so sorry about that. But you can see the Chinese character here and there. So micro needle itself is normally we can say it's a length is something like one less than 1 milliliter, 1 millimeter, and the end of tip sharpness is something like smaller than 50 or 30 micron. Yeah, that is giving no pain is a needle. So that kind of micro needle we should say micro needle, right? So today's talk is I talking about a lot of the only micro needle stuff, but that means a micrometer size needle made of various biocompatible materials. So mainly I also using for all biodissolvable that kind of polymer mainly. So that is can create a pathway into the epidermis or dermis even layers to transport the drug molecule. That is mainly so far as the microneedle society is doing investigating research, and that is minimally invasive, so no pain patient friendly, so non-invasive. Also, no medical professional. That means no medical staff required. Therefore, by yourself in the your home and anytime you want, you can you can deliver the drug. Right? That is very convenient. Also in the stories as well, and it can be used for the vaccination delivered. It is also very strong benefit. You don't need a cold chain, which means refrigerator storage. By the way, so that is conventional microneedle so far, so using especially for drug delivery application. So there are different type of microneedle actually. So I should say thanks again the big boss, our big boss needle. There's a Mark professor here. Is this figure from his paper. So for conventional drug deliveries, microneedle are mainly four type we should say. There is one is solid type microneedle, and then the other one is also the almost same solid type microneedle, but it's the API is a coated one. And the other one is also a hollow needle, which is really miniaturized of the conventional injection needle, right? There is also arrays type. But recently, nowadays for DDS application, drug delivery system application, mainly now is a biodegradable needle, most popular and actually very hot issue. So you can see here some pictures. So the silicone or metal actually is not biodegradable or biocompatible, even something like also sometimes brittle. But it's thanks to the micro technology, so it's based used on the silicone semiconductors lithography way, you can make now the solid type microneedle array very easily. By the way, also it is just first to penetrate the skin barrier and then drug delivery inside without remove this needle patch. By the way, the coated microneedle, this actually is also I should say is most なんていうんですかね、for example, is commercially available for the real some drug, painkiller drug is used that kind of coated microneedle now, and it is another way. By the way, also thanks to MEMS technology and many microfabrication technologies, hollow needle actually did that kind of hollow needle array for the extract inside biofluidic through the skin, and for the sensor application, actually, Professor Boris also very pioneer the scientist about that kind of needle. Well, so there are some depend on the some application. There are something kind of that kind of microneedle technology is coming out, but I should say at least my group and also Professor Mark also starting challenging for the second generation of the microneedle patch, that is which is a dissolvable microneedle. So it's a biodegradable polymer. So that polymer can be fabricated somehow in the microneedle shape, not giving the pain. And then you can see here, so there are several kinds of biodegradable or biodissolvable even polymer made used for that kind of microneedle patch. For example, water soluble polymer we can use in CMC or hyaluronic acid, or there is a maltose here, and also PBP or PBA, etc. And even non-water soluble polymer, but biocomfortable the polymer, sometimes using chitosan and PLA, the polylactic acid, or PLGA, etc. So some group also working some water oil emulsion polymer using for making some shape, something like that. So when you can make some that sharp and small microneedle arrays with that kind of somehow this needle shape and strength, which have enough strong mechanical stiffness, can penetrate the skin, and then uh, uh, you can put many active compounds, something like the hormone, peptide, or DNA even, or some vaccination and vaccination, etc. So you can see here, so that is already existing conventional or drug delivery microneedle patch, something like that. The hydrocolloidal patch, and then there are a lot of microneedle array which made with something like for example, hyaluronic acid, yeah, the which are water salt dissolvable the polymer. Then that the needle penetrates the skin barrier, corneum, and then diffusing delivered to the epidermis. So you can see here already you can buy very easily now in the drugstore commercial available microneedle patch for actually it's a cosmetic field. So to remove wrinkle in the face for anti-aging cosmetic market, actually already since 10 years, hyaluronic acid based microneedle patch with even including the epidermal growing factor or some cytokine inside, also are already available, right? So for example, so just really some starting point of peak of the COVID-19 the pandemic situation in the world, the 2020 there is the World Economic Forum, and they talking about the top 10 emerging technology of 2020. At that time, the number one is actually the microneedle for painless injection, even for test for the self-test for the sensor. I guess it is also very still very have a very big potential to revolutionize our industrial drug industry or healthcare and society. Right. So today, so microneedle talking from my lab. So basically, our needle is all kind of needle is basically the biodiesel polymer based microneedle. So don't forget to until final of my talk, I give 7 topics. So first, number one, we also try to make some microneedle for drug delivery. So poke and deliver. So deliver active pharmaceutical ingredient inside the skin with a non-invasive or effective way. That is I already talked in general talk. So keyword is vaccination. So so many microneedle society and people to try to now tackle this microneedle patch for useful for the real vaccination delivery. So without any cold chain, it can be delivered stably, so much long time, and also well, so in a very warm country like Africa or Southeast Asia, the still vaccine is exists, but they cannot deliver it to the reach the people easily, not easily. But that is for the what cold chain system. Yeah, we need a refrigerator. By the way, we also try to now is still in developing so very new type of COVID-19 vaccination using our microneedle patch. That vaccine is developed in our University of Tokyo and Tokyo Metropolitan Medical Institute. They're using the vaccinia virus as a COVID against COVID-19 vaccine vector. So it's DNA muted and very stable vaccine about the vaccination with COVID-19 now. Therefore, we want to try to make that kind of vaccination with our drill needle. By the way, also we tried to do some different type of micro droplet based some hyaluronic acid or here as a pyrogel based polymer microneedle patch for the drug delivery. But in that case, we want to try to do some more so sustained time, right? So we have several target now is for antibiotic and some curing of the sclerosis disease, skin disease. So using some several aptamer based microneedle patch is still developing. But well, there is this drug delivery system already many people are going and research are going. And my main talk is actually the third target of the needle story, but later I will say. Well, that is 2000, 2003. Yeah, that I was first assigned the professor of University of Tokyo at the time, 2000 or 2023 years ago. As you know, my major is actually silicon-based MEMS fabrication. So the MEMS, you know, the micro electromechanical system. So that time also I working over more about the biosensor from precision machinery department of University of Tokyo. So just when I was first assigned a young professor in University of Tokyo at the time, 2000, my research field basically silicon-based the MEMS. So my first some student with studies here you can see here. So microneedle, that is also actually microneedle, but silicon-based solid needle, and it is not drug delivery. It is electrode. So it is for the neural signal detecting neural probe. That is micro. Those a kind of course, measurement tool in our group. So, but it's minimally invasive micro scales sharp silicon needle array. And even it's a flexible substrate. So that is what the signal detecting. So you can see here some silicon-based needle array with the electrode in the polyimide flexible substrate, and we did many silicon etching and lithography, etc. etc. So finally, we can open the brain of the rat, and then we put our electrode in the brain tissue of the rat, and real time we are finally successfully detected, measure the neuron signal through the multi-area microneedle electrode. But that is my why I should say showing this slide, because still at that time microneedle in for our MEMS guy field, there is something like that kind of neuron probe as for me. But and then I know the microneedle is another good application for the DDS, especially based on the micro needle based with that kind of polymer, etc. There is actually I remember is first time I attended this microneedle conference in the 2000 I can 2020 at the time. This is my first time, and then 2014 I guess is Baltimore, and at the time I saw the microneedle conference I attended. So I know this. You can see here is a very our big boss Mark Baranchi professor is really starting pioneer of this microneedle field for the drug delivery. By the way, that means I should say this several conference continues. So now this year, but just 3 weeks after, we'll get together in Seattle in America. There is a seventh of microneedle conference. That I should say, you can see already feel the only just tens of years, 15 or well, I should say 20, 13 years. It's really starting. So this microneedle, our society for the molecular needle array patch, right? Map society is really industrially also all get together. So the fabricator and researchers get starting. So still, I should say, still young research field. So currently, for about the dissolving microneedle patch, yeah, I should say already you can find very easy in the cosmetic field already commercialized in only cosmetic area. Also, small product, few product already commercial available for skin trouble care, acne or care or something like that. But also some few, I should say few, but it's now it's increasing number of micro DDS system, influenza vaccination research, and for some this level for the medical application is ongoing. Uh, but still, we should say about the minimal invasive manners. Still, in sometimes it's still some giving pain. But well, what I should say for drug delivery system, for me is cure the disease. In that case, I should say it's something like some still giving pain to patient. Actually, it doesn't matter. If really giving new solution to cure some disease, for the medical doctor concerning to the medical doctor is pain is doesn't matter for the really it can be secure the disease. That means we the microneedle patch for the some drug delivery, so to in order to cure some disease you should thinking very carefully for the find some application target which cannot be really delivered for the comparing with the conventional injection needle or even the oral the drug, right? By the way, the why, but this dissolved needle is for the very locally the transfer the drug very easy. That is very still very attractive. So, but why is so easy to disperse so nicely in the drug company drug system? There are some limitation. Is before after I will talk in detail. But by the way, some for fabricator the point of view. So still limitation of low cost mass fabrication with the GMP equipment in very clean environment with also arbitrary shape or various dimension is still difficult. So how about conventional microneedle? I should say it is a dissolvable polymer microneedle, right? That how about conventional microneedle fabrication so far? So I should say so almost 95% competitor. I should say it's competitor, sorry, but some another group is mainly using micro molding system. So to consider very well about the ponder the how can mass fabricate the very this small scale of the microneedle very uniformly. That is maybe the proper the optimized way I should say is a molding technology. So the put the polymer and drug and then the dry and then peel it off. That is which is most easiest way, I should say. So many company from Korea as well as in Japanese, some big company, Fujifilm, Cosmetics, or some other even American 3M and Korean and Terada, etc. They're using micro mold. So I should say this micro needle length is at least more than 300 micrometer and some reach to something like some millimeter, which is actually very very big from viewpoint of MEMS based silicon based micron fabrication people. So we can make a easily nanometer scale or some micron or tens of micrometer scale in the silicon wafer with photolithography very easy. But length of 1 million or few hundred of micrometer is very big. By the way, to avoid the painless or very nicely penetrate the skin, you need a very sharp needle tip. There is something like angle is less than 30 degree, and then the length of radius of tip should be 20 or 30 micron, at most 50 micron, which is very very small. Is difficult to fabricate by top-down precision mechanical machine fabricator. Therefore, our this microneedle scale is really some bootleg of scale, both scale, too big for the bottom approach fabricator, micro machine fabricator, or too small to top down mechanical engineer, the fabricator is both scale is at the same time they have it. So, but finally they can make a micro mold very somehow technology mixing and then put the micro needle polymer inside. But you can see already filling the mold, this normally this digital polymer is the viscosity very high. That kind of high viscosity polymer to fill completely inside the mold to remove air inside the mold in cavity is sometimes become difficult. By the way, drying the polymer, you normally using the heat or UV light, something like that, by mainly heating. So you can see here the thermal conductivity of the mold material and polymer needle itself is very different. So sometimes outside is the polymer micro needle is very easy dried, but the inside of a polymer sometimes takes time more. As there is some giving that uniform mechanical stiffness finally of the needle. But also detaching from the mold the micro needle patch, so without breakage of needle tip. Sometimes need a substrate mount or cleaning, but there some that kind of micro needle polymer micro needle is many technologies very becomes stocked and know-how is becomes go and go beautifully. Then now many people are already conventionally aware of that. And then as for me actually personally to starting this micro needle for the drug delivery system, I some starting collaboration some Korean company which investigate some very straight, I should say very interesting way. That is, they using the hyaluronate, not something like that kind of high viscous polymer by the inkjet printing to the substrate with something like the droplet, some diameter of the few hundred micron, like this. You can see the video, and then the other the patch substrate come down and contact and just drawing, right? But I should say this polymer is very high viscosity, and then you can control the speed and the step, and then finally you just dry the totally completely the uniform of the needle mechanism is drying way with the cold air. So they said it's a droplet borne air blowing. They said this method. So we collaborate with that kind of machine. Then just speed up. They can make a same two needle. There is a successful cosmetic field and mass fabrication, but still that way also still limitation of the combine the various type of API component. Also, it is difficult to fabricate various shape or various lengths. It's always changing the parameter, etc. So when you collaborate with that group, we develop another way to instead of inkjet printing the by one dot, we're using the shadow mask and one time the droplet with a much larger area. Or so using the needle type, the dispenser, we directed to patterning of the needle, etc., etc. But well, there are several that kind of the mainly the molding technologies used. Okay, and then there are several another types of exist. And then in our case, also recently we tried to make a micro needle that basically this polymer is also polylactic acid, which is a biocompatible the polymer, but it can be used for the also 3D printer material. So we're using 3D printing. So we made some sub-milli scale of the various shape of the even sometimes which cannot fabricate the very that kind of tip based this needle, which cannot fabricate through the conventional molding way, because when you tip shape like this the knife edge the needle, you cannot peel off the from the mold outside. That kind of a barrel shape can be easily fabricated the 3D printing, because we found some isotropical etching condition to reduce this 3D printed this barrel shape of structure to micro scale. Therefore, we designed the barrel shape. A final goal of the shape can be done using already known parameter of the etching laser in 3 axis direction. Therefore, we can design the 3D printer and then print it with not micro scale in that case, so some sub-milli scale, and then uh, etching uniformly, and then finally drop API can be quoted. So actually, some to realize the various API to deliver the sort of microneedle, in that case, of a much stable way, conserve the this activity of the API. So normally, so quoted one is also available. So that kind of way also we thinking. Well, so I don't dispense too much time for about that drug delivery application. Second one is you remember I will talk three introduce three way. Second one also we tried to making the poke and irradiate. That means we try to make the microneedle for optical microneedle. That means, as I said, the skin barrier of the sphenocorneum, due to that skin barrier, drug higher molecule of the drug cannot penetrate. Therefore we penetrate the skin through the microneedle structure. By the way, light also depend on his wavelength cannot penetrate inside deep, even the epidermis or dermis area. Normally, that is also scattered or absorbed in the melanin component sometimes in there. So also short wave, for example, blue LED, the short wavelength of light cannot penetrate deeply inside the skin. But you go to the skin care, the clinic or the dermatology hospital, several people working about the laser therapy or even for the cancer treatment, photodynamic therapy still used, right? So we thinking about that application with a microneedle, which made with a transparent polymer needle. Yeah. So maybe but there is application is also hair removal and so on and so on. But I should say, well, I should say more about focusing now literally about the photo therapy for skin. Well, first time we tried to the blue LED as a light source, and then the transparent micropolymer needle with combined with the lens, flexible lenses, polymer substrate. Then we put the blue LED directly through the needle, and then melanoma cell, that is kind of skin cancer cell, also can the killing about this melanoma skin cancer cell is due to the blue LED light also excited, enhance the uh, increasing the なんていうんですかね、I forgot. Anyway, apoptosis area from the cancer cell, but recently, not only just needle transfer the right inside the skin, that application not only. So we more focusing on the photo dynamic therapy for the skin cancer treatment. So I should say that in Europe, one thirty, so something like 30% of adult easily have the skin cancer. So then all the stage of skin cancer treatment is also very urgent and problem. So therefore, our optical needle have been proposed to enhance the subcutaneous light transmission, as shown in this figure, right? And so therefore, optical needle pierced the skin area as well, and transparent needle body transmit the light to the deeper layer of the skin, yeah, and also improving the efficiency of phototherapy. So first of all, we proposed the optical needle lens array as well. So we should say OMLA. But at the time, it's still we working so for that, but still the shape and thickness to mass fabrication for efficiency, and we more also to difficulty of this alignment and actually, and somehow how can focusing light on the end of tip, etc. So we control this dimension and better light transmission performance using mass fabrication. We try to work in the simulated. I should keeps all in detail because of the time. So finally, we made a PLA transparent polymer, and a lens also same thing, and we tried to make some mechanical alignment way, and to also optimize a substrate between microneedle and lens substrate, quite good thickness for the proper thickness to this embossing process to write is more and more giving to deliver the end of tip as well as a microneedle array itself also diffusing very brightly. So there is successfully done, and you can see here light transmission test using porcine skin and conventional just to optical microneedle lens array, the inconsistent thickness of substrate. The comparing that by our previous work, we are control the thickness of substrate of needle and lens designed, and is almost 135% is increasing the light transmission ability. That means it can be using further some any kind of wavelengths of light can be without pain. Just stick to this lens optical needle substrate in the skin, and then so outside of some light source you can penetrate the skin. Well, also for I should say I said about the photodynamic therapy for the skin cancer treatment. In that case, we're using the photosynthesizer. So the photosynthesizer is depend on some several kind of photosynthesizers to light the trigger this photosynthesizer, and that is that kind of photosynthesizer is of course it should be on toxicity in our body, and then when you put the light proper light wavelengths of light in this photosynthesizer, it reacting with this photon, and then they make a radical of the oxygen and sometimes some different some chemical reaction inside the skin derm, and then they can kill the cancer cell, right? So of course, normal cell can also absorb this photosynthesizer, but that is sometimes a higher molecule of that kind of thing. This normal cell is just leave out this photosynthesizer, but cancer cell is like some pig is eat everything, so it's absorbing this photosynthesizer for a long time inside the cancer cell. Then we trigger this light through the needle patch inside the skin. Then it's activated as photosynthesizer and can selectively target locally the treatment of the cancer cell. So well, sorry. So first one is general talk of drug delivery for the drug delivery system, microneedle patch. So many people are very now competitively working. And second one is optical needle patch. It's ongoing, relatively new for our research group as well. And third one, that is actually, sorry, almost 30, 40 minutes already passed. But my main topic actually is not drug delivery. For us, is much focused on the sense application. They mean you can see here some this report and papers after you can see find. So our mission is we believe of course cure disease is also very important, but nowadays as from now is more cure medicine is more changing paradigm shifted for the preventive medicine, right? So prevention is better than cure, but of course cure is much more important. But that preventive medicine is very important. Means preventive solution should be really conveniently and accurately and every day can be we can reach for us. Yeah. So prevention cured. That prevention then means monitoring your body condition, right? That means you need some sensor. That sensing data is the exist means already remote medicine can be done. That is all data transformation at the data should be directly connected to digital healthcare, and this can be helpful for the AI and deep learning. So it's many another some explosive for the new generation of the medical system. So which is important is I should say prevention medicine. That means measuring people is important. There are several stages of the concerning hierarchy of accuracy here in our healthcare management. So you can see very, of course, in the hospital, very nice, precisely motion of uses. So I don't say about that kind of medical instrument measurement, but already wellness or even some just fitness, we already available in wearable sensor. Yeah. So in that case, still wearable sensor in mainly still our motion tracking using the MEM space, the gyroscope or accelerometer, and then also the tracking, also several physical sensing, such as the heart rate or so cardiogram, ECG, and some electrode and measurement and our body temperature, etc. But it is, they said in healthcare, of course, this kind of our motioning and every day, that kind of also temperature and by the skin electronics, you can measure several biomarkers. It's of course very important. So by vital measurement, but really so far still good existing actual point of care tester for real diagnostics. You can see here the pregnant tester is a immunoassay, right? But so through I should say new なんというかな、母親子はピピね。So urine ね。So that kind of thing is exists. So I should say, of course, it's that kind of thing also the glucose sensing with some urine, right? That is already exists, but it is very simple way. But I should say, really, our body bio monitoring with the biomarkers detected very well. This still only one solution is a blood sampling. So when you go to hospital by the blood testing, then okay, so already time. So that is still play a major role in point of care, yeah. But it is still, you can see here, yeah, the still giving cause the pain and also discomfort for patients every day detect who should detect the glucose, for example, glucose level. So therefore, yeah, rapid painless detection. Well, I should say in our cases within 2 5 minutes by naked eye using colorimetric sensing. It depends on the concentration of our biomarker, of which is, for example, glucose or cholesterol, and also we now working about cortisol, which is a hormone, stress marker hormone. Yeah. So that kind of all the component is actually in our body. Especially I should say in later it's interstitial fluid that is increasing. Therefore, we observe, I should say, extract that our body fluidic instead of the blood without pain easily, and then that is coming out through our microneedle sensor application needle, which is actually porous polymer needle, interconnected porous needle. So and then detecting that depend on this concentration of our biomarker. We put our paper sensor is we are selectively patterned of some several enzyme which react chemically reacting, and then which contain also coloric ingredient. Therefore, depend on this, which is easily over the conventional normally is using for that kind of tester. So, depend on this intensity of the color changing of the sensing area. So, I should say. So, I already said there's our target through the microneedle absorption beyond the skin, that is interstitial fluidic. So skin interstitial fluidic, ISF, has become a good alternative to blood sampling due to this broad body containing an abundant source of biomarker, which is accurately reflect their concentration in blood plasma. So compare with the saliva or sweat or tears, for example, is in the case of the glucose, its concentration. So you can see ISF is plasma almost same. So therefore we focus on interstitial fluid as a biomarker because 95% of its constituent in component are the exact same as the blood plasma normally used for such a sensor monitoring, right? So it's the Abbott company is now is developed a 2 weeks continuous monitoring of glucose sensor. There is put the micro electrode in the through the skin and continuous monitoring of the glucose level. That is in that case, continuous 2 weeks just stick to your skin. That is, it cannot be penetrated even the blood vessel. So there are in that case also interstitial fluid is a target, yeah, bio target. So I say so. Our third topic is a biosensor microneedle patch. Is a poke and extract and analyze. So we are targeting interstitial fluid and analyzing ISF for sensing or monitoring the change of bite function. So it's a really fast and simple diagnostic site. So it's a really good key component for the biosensor. Yeah. So far we working on colorimetric sensing. So our target is, so you can see here, it is a glucose sensing, and also of course there are ISF is contain also or various kind of the protein and enzyme as well. So you can detecting antibody as well. So, for example, we successfully detect with this our homemade lateral flow test paper-based sensing area as well as a microneedle behind this, so porous microneedle. So basically, our sensing application microneedle made with polymer, biodiesel polymer, which is but have to have the interconnected porous channel. So therefore, actually, this one is also actual the picture, experimental picture, depending on the cholesterol. Or of course, cholesterol is a nice one, nice guy and bad guy. You can say the HDL and LDL also exist, but in our case, the general total cholesterol level also can be detected by the colorimetric sensing, right? So for example, the glucose sensing, we put the two kind of enzymes, cholesterase and paradoxase. So the depend on concentration of the glucose level inside the interstitial fluid or in body fluid, they're extracted and then combined with activated these two enzymes, and then there are hydrogen element coming out. Then the paradoxase is activate, and then that is also combined with, we put the color sensor, color components, which is a TMB. I maybe will be shown in the later in the in the afterward. Okay, I skip here. Okay, you can see here the porous polymer needle exists, and the directly fabricated in the paper cell. Paper is also very nice, organized, connected, interconnected porous structure. The cellulose fiber and nano channel. So, and then opposite side there is here you can see here TMB. There's glucose to glucose oxidase is to oxygen, and the gluconic acid to divide the H2O2, and the paradoxase enzyme is activated with the injection that is going out the electron, and then TMB molecule can be changed, the TMB oxidized, then it can be color is changing. So TMB also conventionally used for the colorimetric components. So we try to do so much. That is very old data, but now it's more stable and more secure, accurate color changing can be detected by the handphone or just imaging software. You can, of course, digitally can data is obtained. By the way, the color intensity depends on the glucose concentration. It can be changed, right? So this almost 150 microliters, or actually a lot of that is can be within 5 minutes. So our array of the microneedle, porous microneedle, can be directly observed. Okay, so maybe I should going more further. The previous one is how can make it. So first time we using the salt treating method. So this the polymer and then salt, the salt is also, you can buy various diameter of the salt powder, right? There is something like sometimes a 10 or 20 micrometer in diameter. the salt and mix the polymer, and then in the mold you can make a shape, right? And then you can put just the hot water. Then if salt is interconnected somehow, that is within what water is some dissolved, right? And then they make inside the interconnected narrow channel, right? Then you can make by capillary force, you can extract some biofluidic through this porous needle, right? And that is our first time trial. And then, but you can see here the slide. Nowadays, we tried another way. So because salt reaching is very difficult to control the fine porosity, yeah. Also, mechanical stiffness also difficult to contain. Therefore, since we're using, for example, we using this fluor focus micro devices. So you made the micro droplet shape of the polymer, and then just mixing inside the mold, and just some heat treatment. Very easily, you can get a very control over the porosity and this interconnected structure. Even this needle itself also very mechanically stiff, is strong enough to penetrate without any breakage of needle, right? So first target is for the I should say the prevention medicine. So we pre-detected this disease early stages. One of our target is, as you know, is diabetes. Yeah, the I should say is almost nowadays. I should say is well 1 for 11. So 1 in 11 now in the world population are living with diabetes. It's a very big problem, right? And then type 2 diabetes increasing a lot. So that is also important issue. Type 2 in diabetes increasing means type 2 diabetes is prevent this diabetes type 2. You should take care of the early stage. So take care of this become diabetes disease. That means uh, already exists many people have a pre-diabetes situation. So I should say this said that 1/3 of world population, by 30% of adult is pre-diabetes. But much problem is you can hear right here. You can also much problem is of them, these pre-diabetic people, of them, 80% are unknown on where they are already pre-diabetes situation. So just 2 times per year, when go to the hospital and healthcare testing and blood testing, the doctor said you are today is very high cholesterol level, but glucose level. Then sometimes they can answer, but you know, so every day is the glucose level is changing always. So you cannot detect, or you cannot even changing your life daily life of food or exercise, physical exercise, that kind of customs cannot be difficult to changing. So, but we should pre-detect of that kind of early stage. Therefore, we need some very low cost and also an accurate and painless that kind of detecting biosense is necessary. So you are our agreement about that, I should say. So we try to make the porous needle for that kind of extract biofluidic and then to detecting all the stages of several the concentration of the biomarker. So as I said, comparison with the previous study on glucose monitoring described in this slide. So many studies have been reported. So replace the conventional device, something like the self-monitoring kit, or even some continuous monitoring kit. The commercial are available, and there are many people working about the tears and the sweat by the electric tattoos, the electric strain, or sometimes the metabolic heat conformation changing the depend on the glucose levels, or some telemetry with broad but the target is broad, but telemetry but optically measure the glucose level, etc. But also urine test also already used for, but it is only for diabetic patients because urine target is a very low level of the glucose level itself compared with the blood plasma. So if you are diabetic patient and your focused treatment is necessary, guy, normally is your high glucose level, it can be detected. but I should say we are targeting more about the pre-diabetic patients. So for daily using, therefore, we concentrated just point of care. You can using this very easy. The painless sensor is 4 or 5 times in the day and checking, and then it's just the color changing. You can take the photo with your cellular phone, and there is image tool. You can digitalize the data. Of course, you need a compensate なんだ、プリアの、ね、テストね、やって、それで、when you see the also the limit of detection LOD of through our columnar sensor, because of using the I would say interceptor fluidic targets, they're very precise. 0.1 to millimole can be detected only for the columnar sensing. Also, we try to do continuous monitoring as well as in that case much complex, and our sensing method is not columnar sensor. We should be using for the electrode, but thanks to the silicon-based MEMS structure with the capillary pump, you can see here the video. Yeah, you can this capillary pump can extract the micro. You can see that this fluid is coming out. So continuous 6 hours to completely contain the microfluidic device. You can continuously pull out extracted also the microfluidic device through the needle. Okay, this is also another very interesting showing. So we are actually with the 2-photon 3D lithography. We very nicely, this actual same image of our productive porous needle structure with polymer. It's hydrophobic, but it's a very well controlled volume of the porosity of the cavity is the fractal structure. Therefore, only by the capillary force, you can see even hydrophobic polymer, you can extract it, exactly define the nanoliter of this volume of the liquid can be extracted, and then you can put in the artificial skin. It's hydrophilic, then it's very easy just dispute this deliver the drug, because it's it's without any pumping. You can that kind of nanoliter pipetting can be done. But I should say this just for the showing the what I should say is for the capillary pump, or how is this important and interesting. So finally, I have some slides for the COVID-19. The antibody detection can be also done with our porous needle and this conventional this lateral flow tester. We can finally very nicely detect the COVID IgG, I should say that kind of IgG or IgM, the antibody can be detectable. Well, thanks a lot. So I almost time for the finishing my talk. I should say, ooh la la. I should say this. Our dream is that kind of flexible, just some sensor in your clothes to detecting daily time through the skin, detecting your body marker. That is our final dream. Okay. Okay. Thanks a lot for your attendance and for audience. So it's my final talk. Thanks a lot.
Thank you very much, Professor Kim, for such a wonderful talk. Yeah, I think you just hit the right time. And now let me introduce our panelists tonight for wonderful discussions. So the first, today we have an extremely strong panelist teams. Our first panelist is Professor Mark Proutsky from Georgia Institute of Technology. Mark is a Regent Professor and also Regent Entrepreneur. He earned his bachelor from Stanford and PhD from MIT, both in chemical engineering. And Mark and his colleagues carry out research on drug delivery using microneedles. As Kim also mentioned, Professor Kim also mentioned, it's kind of the pioneer for the field. And also using lasers, ionic liquids, as well as other micro devices for transdermal ocular and intracellular delivery of drugs and vaccines. Mark teaches courses on engineering, calculations, and pharmaceuticals, and published more than 300 journal articles and co-founded 8 startup companies. Wow, that's really amazing, including Biomedicals, Adena Therapeutics, and Clearsight Biomedicals. Our second panelist today is also quite famous in the field. Is Professor Boris Sturber from UBC. Boris received his electrical engineering diploma from Germany in 1998 and general engineering diploma from France, and also PhD in mechanical engineering from UC Berkeley in 2002. And from 2003 to 2005, Boris is a postdoc scientist in chemical engineering at the University of California, Berkeley. And afterwards, since 2005, he's with University of British Columbia with the Department of Mechanical Engineering and Department of Electrical and Computer Engineering. And Boris' research interests including the flow physics at small length scales, microflow imaging methods, micro-optical devices, sensing technology, and medical micro devices, and fabrication techniques for microelectromechanics MEMS structures. And also, he has the co-chairs of the 5th International Conference on Microneedles, and also co-editor books, and also guest editors for the special issues in the field. And today, we also have our ex-challenger, Ms. Jiayan Zhang, who is a second-year PhD candidate in microelectronics and solid-state electronics at Peking Universities, under the supervision of Professor Zhihong Li. Ms. Zhang received the bachelor's in microelectronics and solid-state electronics from Eastern China Normal Universities in 2021 and continued her doctoral studies at Peking University. Her research interest is in biomems, including the design of microneedles, electrode, and its application in the electrophysiological signals recordings. And she also received many of the research grants and also many scholarships. And also, today we have Martins with us. So everyone today basically have the background with MEMS, with microneedles. So now let's welcome all the panelists, ex-challengers, and moderators on the stage for the discussions. Okay, so as usual, our first turn is for the ex-challengers. So Jiayan, the stage is yours. Please ask questions.
Okay, thanks, Professor Chen, for the introduction. It's a wonderful talk, and Professor Kim have shared a lot about the microneedles. I really learned a lot. Thank you, Professor Kim. Here I have 2 questions. First, could you illustrate more about how long it will take to finish the detection of biomarkers when using the biosensor microneedle patch? And the next one is, what do you think it will be the challenge in the application of microneedles? Okay, that's all my questions.
Professor Kim, please.
You're muted. Sorry. Okay. Thanks again. Ms. Chang. So very good question and very interesting question. So first one, Professor, you say about how long it takes time to finishing this sensor, to extract biofluidics. So that is depend on the how many microneedle, how many can extract the biofluidic, to enough detection volume. But I should say there is takes something like 2 or in our case, micropores needle array with that is it takes something like 2 or 3 minutes to extract enough sensing volume, which is 100 microliter, is coming out within 5 minutes, within 2 to 3 minutes, yeah, normally. And then it should be react enzyme so to giving reach the saturated color density changing and depend on the inside the ingredient the cholesterol biomarkers the density, yeah, I should say concentration. They take something like normally 1 or 2 minutes. So totally 6-10 minutes you can get the result. It's good. Is the answer. Yeah. That is columeric sense.
Thank you.
By the way, so what is challenging for microneedle? There's a lot of challenging. So it is more good 100% good answer can be obtained by our boss Mark. By the way, for drug delivery, I should say before I said we should targeting is a very good targeting ingredient for the curing disease or helping or anti-aging. There are all the stage they working over the growing factor, can be hormone, can be used for vaccination, etc. There are some specific advantage should be obtained for the microneedle for drug delivery. By the way, so many microneedle drug patch, even drug delivery or sensing area, we're thinking about the very uniformly to penetrate in the skin, right, without pain. In that case, we should design proper the needle, the density and gap and shape and the length, etc. By the way, also, well, uniform force should be the necessary. That means applicator, proper applicator or some good you to consider, right? Real the commercially available. Also, recently I saw also, for example, markers for cosmetic area, they don't using the patch, yeah. Just some they said star, so something like a microspicule, that kind of needle component itself structure is combined some cream, right? And then use for the that is a microstructure in the just the inside ingredient in the cream, cosmetic cream, for example, then can very efficiently very small pain there is the skin and then penetrate the good components inside the skin. That application is also available, right? They're promising. There is already recently Professor Mark already published. That depends on the goal. So even microneedle patch array should be considered several parts. So safety, the material issue, and so good target for the advantage using that easily available. The pain is the advantage can be should have some real advantage. Yeah. So but sense application, yeah, is also still starting point. So Professor Boris is working also have also developed a company using the in our case porous needle, right? But their group is a hollow needle. Yeah, that is also very good applicate for extract biofluidic, yeah, very easily. Also very not doing this kind of 100% completely, yeah, really coming out. So in that case, also pumping system. Of course, he using also narrow structure. Therefore, he using also capillary process, etc. Of course, but in that case, the material issue. How can without breakage of that kind of metal or some material, that is also issue. But I think it's a yeah, motivation and application is very from now is very promising and large. But several the product issue you should take care of several things to have the real permission from the FDI. Yeah.
Yep.
FDA, yeah.
FDA, yep.
Yeah.
Very, very good, very interesting. And I would want to ask Boris and Mark to jump in if you can, because the questions that our externals ask are very stimulating. With the technology, as you described, so much is happening. Definitely the interest is there. So what do we need to do as a community Do we need to get other people from other fields to come and join join forces with you? Do we need to maybe even talk more about this microneedle and really push to get them to a point where medicine doesn't have to be so complicated and so invasive? Boris and Mark, do you have any comments on top of what Professor Kim said?
So I could, yes, I can offer some thoughts. So I think the presentation was really notable in that it covered many topics, right? The concept of a microneedle is a very simple concept, but as we saw in the talk, it can manifest itself in so many ways. You can use it in so many different ways as this minimally invasive interface with the body that you put things in, you take things out. There are many possibilities. So when I think about how to go from the many possibilities to realities, to actual products that can be used in medicine, I think that there are two key steps that have not happened enough. And so one of them is moving into clinical trials. So it's much easier if you have an innovative idea, you can do a preclinical study, you can get it published, you can get people excited, but to get into the clinical trial is quite expensive and a lot more work. And actually, many of the researchers who are doing the more basic research don't do clinical trials. And so it needs collaborations and teams and so forth. So I see that as an important limiting step for pharma and other companies to get interested. They will be much more interested with clinical data. And so I think the more clinical data we can generate, the better off we'll be to convince industry. And then the second topic, and this is one that I think Dr. Kim could especially address, is manufacturing. So we can make small numbers of these, and if we don't pay graduate students too much, they don't cost too much to make, but just joking about that. But if you want to mass produce this and make it be useful, you have to have not only quality, but you have to have cost constraints. For most of these applications, you can't have it cost $100 for making a patch. It should many times be less than $1 to make a patch. So that would be my question for Dr. Kim, is your thoughts about the manufacturing. And I was interested, you mentioned there are approaches other than molding that could be considered. And so could you elaborate a little bit more about what you think can get us there in terms of a manufacturing process that will be cost effective and reliable?
Yeah, that is a good question. Yeah, really thanks for Mark. So I should say for us now it's more concentrate for the sensor application we do, which is a porous polymer microneedle. So in that case, you should using only way is a molding technology. So, but to reduce the cost, now we are several film company in Japan is collaborate with us. There's somehow press roll press way. So that is now trying to this molding with some polymer ingredient is some pressing, yeah, like a roll to roll, but not completely roll to roll, some pressing rolling, yeah. That is now equipment is discussed, but and then it's GMP quality, and yeah, the sanitation is very easily done because it's a sensor, so therefore not inside the drug inside. So right, so that kind of thing is now approaching way. So that is our, yeah, I should say somehow it's the answer right direction.
Yeah, maybe I'll ask a question. As was asked, so how do we make people more excited about this field? How do people make more people join? And I think it's definitely as we've seen, it's a highly interdisciplinary field, right? And so maybe Dr. Kim, you can give a few insights on what you think are maybe the fundamentals that new scientists might want to learn, maybe a course that could be developed to make that happen. And maybe I'll start with that. I have other questions.
I see. Well, so this, yeah. When I collaborate with a medical doctor, normally I working with the dermatologist people, right? So then, for example, curing some skin disease. normally there already exists just injection needle, so they don't care about the patient's pain. Yeah. So if you cure some skin disease with a new way, well, with the new drug, there's much for they are interested. So another treatment with using laser or light, something like that. So that kind of thing we discussing with a medical doctor. So in that case, difficult to find the real merit of a microneedle itself. So sometimes some is not good way as well. So therefore, I should say some really clinical based some medical doctor opinion are very different for our microneedle oriented engineer guys. So there is some more and good connection and many people interacted that the field is very important. And also, we also working now with some detecting, for example, female hormone. So femtech is a female technology, one kind of that also in important. So I working with some female medical. I pregnant for pregnant. Yeah, right. That is we also fatality. I don't exactly understand. Remember the English name, but that is also the female estrogen, something like that. Hormone detecting also working. Therefore, some for sensor orientation also we connected to some medical doctor and different field people, not only pharmaceutical people. So, but this difficulties, Mark already mentioned. So when you even the research based some collaboration is done, but still it is for the realized for the commercialization is many so harder for the FDA regulatory system for the safety and material issues to be considered, but it takes time. But I think it's very promising, I guess, for prevention medicine application, microneedle itself. So I'm not sure it's a good question for you, but we need unfortunately we will meet 2 weeks later in the microneedle conference in the theater. So we should discuss many things about that. Yeah, I'm very exciting to discuss with you. Yeah.
Very, very nice question and answer. And maybe I can follow up on that a little bit. One of the most big challenges of anything that is implantable is the material itself. And I saw you making some of the microneedles from polymers, some that are from metals. Is there a material challenge in making these microneedles, or can we sample from a large variety of materials?
Well, so about the material, I guess already many materials tried and many researchers already have been studied. So there is almost several main is hyaluronase, CMC or PLA and PLGA or PGA and several material and already discussed and many tried it. Of course, sometimes some silk fabric also included and many things done. But material itself, I should say, it's not so big, challenging and remain issue. But I should say more about of course, depend on application. I should say several times. It's some just different direction can be considered, but mainly for the real next step of microneedle, kind of combination product. It is not drug. It is not device. It's or not biological product only. It is somehow it's a combination product as a regulation issue. That means we need some standardization of this microneedle patch array. So for example, risk analysis should be considered for injury. It's a really good safety issue in our body. Also, when you want to deliver really drug, in that case, how can control the dose, exact dose or underdose or overdose should be considered. Also, I should say, so material side we should consider biocompatibility and sterility or cleaning, etc. But I should say, some material itself is already many studied. So some typical conventional material is easily fabricated, and the stable one is already many studies done, I guess. Yeah. So I should say since 5 or 10 years, it's increasing, exponentially increasing now. It's all over the world. It's a microneedle issue, I should say. Yeah. Also, the main for drug delivery, I should say is main target is still vaccination, because it's a very big market as well as very important, and also you can definitely get the advantage with without cold chain delivery using through this microneedle. So Mark already almost phase 2, or I should say he already developed it with a nice applicator. Is for the influenza vaccination already done. I guess it's dramatically we have some revolution in drug system in near future, I should say.
Very, very good.
Yeah.
Any other comments from the panel?
I could ask another, make a comment and question,
Yeah.
and that is in the area of diagnostics and of sensing. And I think there are at least two kinds of approaches. One approach, which I think is the one that you emphasize, is where you make measurements in situ. So, well, actually, maybe there's even 3 scenarios. So in any case, one is you make the measurement in situ. You build a sensor capability into the microneedle. You place it on the skin, and then you make those measurements. And that is similar, although scaled down to a microscale, to the only thing that is measured in interstitial fluid today, which is the continuous glucose monitor. It takes a sensor on a much bigger structure, puts it subcutaneously, but it's widely used and successful. So the microneedle allows you to make this a much simpler interface and do those measurements. The other approach is the way almost all other measurements are done, and that is you take the fluid out of the body, and usually that's blood, and then you measure it in some kind of external device. And so you could do that likewise, pulling interstitial fluid out. So do you have some comments about how you decide which is the right approach, to build the sensor into the needle or to pull the fluid out and do the sensing externally?
Yeah, this was a very good question or comment. Thanks a lot again. So for glucose sensing for pre-diabetes, already we done is colorimetric sensing, as you know. It's generally in situ, yeah? In the paper substrates that the enzyme and colorimetric color changing can be. We can just naked eye, you can detect it, right? The in point of care system. So it's disposal and one time measure. It is okay. But I should say for another some biomarker, something like hormone, etc., it is also limited of volume inside already in fluidic. So for in our solution is through our microneedle, just take off that interfluidic which contain the small amount of this some special biomarker hormone, something like that, and then this should be delivered to another some clinical center, yeah, by some which has already nice immunoassay machine or there. So we just dissolving or some centrifuger, there's extract our human body extracted the sample, and then they can measure some that kind of service system also can be available. So, but our motivation is we don't make a very complex system like electric signal and the transfer, etc. It's a very still hard another hard way. So we more combine with the conventional measurements instrument and our microneedle only for the tool for the easily extract the biofluidic. Yeah.
Yeah. May I ask a follow-up question?
Yeah.
Sure. Yeah. So thanks for presenting your exciting work on these porous needles. And you talked about first using them for this poke extract analyze, what else you just outlined again. But then also you talked about using them for continuous sensing.
I see.
There was a time limitation, so you could do it up to 6 hours. And I was wondering if you give us some insights on what issues are or difficulties associated with this, and maybe how you could extend this application.
Yeah, there is so sensing way is not only the enzyme and some the colorimetric sensing. Not only, of course, there exists the electrochemical sensing, right? So they put in the electrode here, and then this microneedle. But in that case, we need some microfluidic chip as well. So we made for example, this one. So we made a porous micropolymer needle is combined the inlet chamber in silicon chip, and there is a microfluidic channel, and the middle space there we should put the electrode for AGAGCL, the reference electrode for continuously monitoring this fluidic, so the fluidic, the biomarker. And then that is another one is open capillary pump. That is thanks to the microstructure in this chamber. There in until completely the fluidic is fill out this space, they are continuously can extract through the skin the fluidic. But in our case, it is really the optical image of our device, the silicon base. It is just we put the color, but it's a real picture. That is a 5 milli here. You can see it's a very few centimeter chip is necessary, and this should be also electrode should be patterned here, and then well, so as I said, it's in the conclusion, it's becomes much complex system, but it should be also necessary somehow. Therefore, we are now thinking about well, I should say it's not only electrochemical measurement. Actually, we are Professor Takeyoshi, our colleague from University of Tokyo, he developed some fluorescent polymer, which combine the glucose level. This is fluorescent is light coming out. That biocompatible polymer, we put our optical microneedle patch and put, and continuously this at least that is they is stably activated this polymer with glucose level, and they can extract the light, fluorescent light. So there is another way to continuous monitoring. So well, so it's still many space to develop and advance the sensing way as well.
All right. I think we definitely have such a wonderful discussions. It's really a time, and thanks again, Professor Kim, for such a wonderful talk tonight. We all enjoyed it, and yeah, on behalf of the ICANX tonight, we want to really present you the electronic certificate to show our appreciation for the wonderful talk. And yeah, definitely hope to see you in person next time. Yeah,
sure.
Yeah. So just to wrap up, a little bit advertisement for next week. ICANX Talks 142. We, as usual, the first week of every month, we have the Young Scientist Awardist, and we're going to have a very good night. And basically, for I hope everyone will be stay tuned for next week. And for now, thanks everyone. And also for me here, it's good night for everyone. Thank you.
再见，你辛苦了。
Hi.
不再是奇迹，不再是幻想，此刻真感觉全世界对我鼓掌。不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱。我创造奇迹，我拥有梦想，我希望看见所有骄傲的人旁。再为曾经失败放弃或感伤，努力才是真的方向。I can， 没有什么可以阻挡心中 无限的力量。I can I can， 你也能够像我一样飞越最高山岗。I can I can， 没有什么可以阻挡心中无限的力量。I can I can， 你也能够像我一样飞越最高山岗。我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞，不再为曾经失败放弃或感伤，努力才是真的方向。I can I can， 没有什么可以阻挡心中无限的力量。I can I can， 你也能够像我一样飞越最高山岗。I can I can， 没有什么可以阻挡心中无限的力量。I can I can， 你也能够像我一样飞越最高山岗。能够像我一样飞越最高山岗。
来，干杯，走！哎，那盘菜不错的样子啊。
有眼光，我们离开电子元器件啊，都没法生活。这个电子元器件是哪里产的？
我知道，是太阳诱电的。
太阳诱电？没听说过呀。
什么？连太阳诱电都不知道？我可一直都是太阳诱电的粉丝。
要说电子元器件的话，还得是和。
不，太阳诱电也是行业的大佬之一呀。高可靠性、高性能，非常棒的电子元器件跨国企业。亏你们也是高新技术圈里的，连这个也不懂。哎，别光顾着吃啊，再听我说话吗？汽车夹什么北京烤鸭啊？你会吃吗？要吃也是吃太阳诱电的吧。
不再是奇迹，不再是幻想，此刻真感觉全世界为我鼓掌，不必太在意身旁惊奇的目光，可以点点头，可以放声歌唱。我创造奇迹，我拥有梦想，我希望看见所有骄傲的脸庞，不再为曾经失败放弃或感伤，努力才是真的方向。I can I can， 没有什么可以阻挡心中无限的力量。I can I can， 你也能够像我一样飞越